Hello guys, welcome back to the Gauravyadavchannel channel. Today we are gonna learn a new topic which is Spirochetes. So in Spirochetes today we are gonna discuss about Treponema which is a causative agent of syphilis. So let's start our lecture. So before start talking about the Triponema I would like to tell you about what is spirochetes. Spirochetes are the elongated flexible bacteria which are twisted spirally along the axis are known as spirochetes. The word spirochetes are derived from two words. One is spiria, which meaning is coil, and second is chet, which meaning is hair. And the characteristic feature of spirochetes is the presence of varying number of endoflagella, and the endoflagella can be polar in nature. The spirochetes are gram-negative and size varying from 5 to 100 micron. They can be aerobic, anaerobic, and facultative. And the example of spirochetes are Treponema species, Borrelia species, and Leptospira species. This is the comparative morphology of spirochetes, and this is the Leptospira, this is the Borrelia, and this is Spiralum, and this is the cross section of internal structure of flagella, or you can say cross view of Leptospira or spirochetes. This is the flagella, these are the endoflagella, and this is the outer membrane, this is the inner membrane, and these are the spiral slender of spirochetes. The genus Treponema are short, slender spirochetes with fine spiral and pointed round ends. Some are pathogenic, while some species are commensal, which resides in our mouth, intestine, and genitalia. Pathogenic species can be grown in cell-containing medium like testes, or while non-pathogenic organism can be cultivable in artificial medium. So Treponema causes various type of disease in humans. First is venereal syphilis, which is caused by Treponemapallidum. Second is endemic syphilis, which is caused by Treponemapallidum and Treponema endemicum. Third is yaws, which is caused by Treponema pertuini. Pinta, which is caused by Treponema carateum. These are non-venereal syphilis, and this is the venereal syphilis, which is caused by the Treponemapallidum. So now talk about the Treponemapallidum, which is a spirochete of syphilis. It is discovered by Scouden and Hoffman in 1905 from the cancres and inguinal lymph nodes of syphilitic patient. Cancres are the ulcer which are painless and mainly formed in the syphilitic disease, syphilitic patient only. And inguinal lymph nodes are the lymph nodes which are present in the groin area of men. Human is the natural host for the Treponemapallidum. They are extremely fastidious and sensitive, means they require special kind of condition, special kind of media for their growth. Like they do not culturable, they do not grow in artificial medium. They can only be grown in cell containing medium like real cell, like testicular cells. They can be grown in rabbit testiculars. They cannot survive long outside of the host, and they can be transmitted by sexually and transplacental route. And the infectious dose is 57 organism. Means to cause disease, Treponema needed at least 57 number of organism to cause a disease in a healthy human or a non-healthy human being. This picture is of cancre, cancre on the lips, and cancre can be formed on lips, penis, or even in the on the vagina. And this is picture of the picture is of inguinal ligament or inguinal lymph node. Morphology: it is a thin, and size range is 4 to 14 long, and 0.1 to 0.2 micron is wide. It has about 10 regular spirals which are sharp, angular, and at regular interval of about 10 micron. They are actively motile, and they cannot be seen under light microscope in wet films, and but they can be seen in negative staining with Indian ink. Its morphology and motility can be seen under dark ground or phase contrast microscope. This is the picture of Treponemapallidum. This is the axial fibril. This is the lipoprotein layer. This is the nuclear region. This is again axial fibril. This is outer sheet, and this is axial filament fibril, and this is insertion pore. This is a picture of cell wall layer. Like as you know, the Treponema is a gram negative. So the Treponema has trilaminar layer, so which provide organism rigidity and shape. This is the cellular membrane, this is peptidoglycan, this is periplasmic space, this is outer membrane, and this is rare transmembrane protein. As you all know, the gram negative bacteria has porine proteins. This is the LP1, this is EF, and this is LP2. The Triponema species cannot be stained by normal dye, but they can be stained with light rose red in color if we stain Treponema with Giemsa stain. It can be stained with silver impregnation method. Fontana's method is useful for staining, and Lavaditti's method used for the tissue section. Ultrastructurally, cytoplasm of Treponema is surrounded by trilaminar cytoplasmic membrane, which is enclosed by a cell wall containing peptidoglycan, which gives the cell rigidity and its shape. The outer membrane protein, it has 3 outer membrane protein: TROM1, TROM2, TROM3, and inner protein are of 15 kilodalton, 17 kilodalton, 45.5 kilodalton, and 47 kilodalton. These are endoflagellum, cytoplasmic membrane, and cytoplasmic inner protein, and it is usually multiplied by binary transverse fission. Cultivation. Pathogenic strains do not grow in artificial culture medium. One is Ritter strain. It has same morphological and antigenic similarity as of Treponema and widely used as antigen in group specific treponemal test for diagnosis of syphilis. Ritter Treponema grows well in thioglycolate medium containing serum, and it is now known as Treponema fagidenis. This is the picture of Treponema fagidenis strain V1, which is cultivated anaerobically on FAA plate and at the 37 degrees Celsius during and for the 8 days. This is the real image. This is the larger image of the colony. Actually, colony is very small in size, as you can see, and the size of the colony is 10 and 5 mm respectively. Second is nicole strains. It is a virulent Treponemapallidum strain which cannot be cultivated in artificial medium. So the one Treponemapallidum strain is maintained in rabbit testicular by serial passage for the decades. As you can see, this is a microscopic image. The growth, spread and eventually destruction of the Treponemapallidum in rabbit testes. ये जो 4 इमेज है, इन एक में स्टार्टिंग से इसमें क्या हो रहा है कि इसमें ग्रो हो रहा है, में स्प्रेड हो रहा है, देन लास्ट में जाके उसका डिस्ट्रक्शन हो रहा है. डिस्ट्रक्शन किस में हो रहा है? रैबिट के टेस्टिस में, जो कि किससे डिमॉन्स्ट्रेट किया गया है? डायरेक्ट इम्यूनोफ्लोरेसेंट एग्जामिनेशन से. फर्स्ट इमेज में द स्मॉल एरो पॉइंट टू फ्यू Treponemapallidum in interstitial tissue of the testis. The large arrow point to the fluorescent red cells in the blood vessels. These are the fluorescent red cells in the blood vessels. The B large arrow point to the fluorescent Treponemapallidum in the interstitial tissue. The non-fluorescent structure are seminiferous tubules, and the C image, which is the day 13, the smaller arrow point to few faintly staining spiral Treponemapallidum. The larger arrow, these are the larger arrow, point to the fluorescent fragment of Treponemapallidum in the macrophages within testicular lesion. And the D image only show red blood cells are fluorescent and no organism can be identified in multiple section of the testes. Now talk about the antigenic structures. There are 3 types of antibodies which form in the treponemal infection. First is reagent antibody that react in the standard or non-specific tests such as Wassermann, Kahn test and VDRL test, in which hapten extracted from the beef fat. What is hapten? Hapten is a small molecular weight compound which is conjugated with carrier protein to induce the specific antibodies, and this lipid hapten is called cardiolipin. Group antigen, second is group antigen, which is found in the Treponemapallidum as well as in the non-pathogenic strains. Second is specific species, and antigen and specific species antigen is polysaccharide in nature, and the antibody to this only found in positive in pathogenic treponemes. Thank you for watching my videos, and please do subscribe my channel. And this is the only first part of the Treponemapallidum, and part 2 will also become after few days. And please do subscribe my channel, like my videos and share my videos, and do watch my other videos also in my playlist. Thank you.